Hello students, in this lecture video, I'll be telling you how biotechnology can be applied in the sector of medicine. So recombinant DNA technology can be used in the healthcare sector for mass production of therapeutic drugs. So recombinant therapeutics, they do not induce any unwanted immunological response, which is the common case when we use anything from non-human source. So that is why around 12 of the recombinant therapeutics are presently being marketed. Now coming to which biotherapeutics are commonly used. First is genetically engineered insulin. Now insulin have been you can say produced from other animals also. Now if we use a bacterium that produces human insulin, then obviously no immunological response will be generated. That is the whole process, so that the thing becomes simpler, and by genetically modified insulin no immunological response is generated in the body. So that is why human insulin gene was identified and then later on cloned in the bacterial cell. So firstly, let us look at the structure of insulin, what all has been discovered. So there is firstly a proinsulin formed, which contains this portion you can see in between portion, this gray portion. It is called as C-peptide. The proinsulin contains the C-peptide, which makes it inactive in nature. Now, when this is processed in the human body, it will form A peptide, that is A chain, and B peptide, that is B chain. So these two polypeptide chains of insulin are joined by disulfide bridges, and this free C peptide is released. So this is the active insulin. So after this identification of the function, there was an American company, Eli Lilly. In 1983, what they did, they took the DNA sequence of A peptide and the sequence of B peptide. Then they cloned these chains separately. After that, artificially, chemically, they created disulfide bonds, and you can say recombinant insulin was produced to be used as a human insulin. Next, coming to gene therapy. In gene therapy also, this can be used in biotechnological sector. Now, what is gene therapy? A therapy which will correct the gene if genetic disorder is there, and if it is identified or diagnosed in embryo, so before birth also it can be rectified. So first clinical gene therapy was given to a 4-year-old girl in which there was ADA deficiency. What is ADA? It is adenosine deaminase. So this enzyme is crucial for immune system. So if this is you can say not functioning well, what is done? A cDNA is formed out of it. Now what is the cDNA? DNA forms RNA by transcription. In some cases, RNA can be reverse transcribed into DNA. That is called as cDNA. So cDNA can be cloned and then it can be expressed, and hence this gene disorder can be corrected. One way is this. Another way is using lymphocytes which are expressing this functional ADA, but then the person has to again and again get those lymphocyte replacement. Or the enzyme which is adenosine deaminase, this is being replaced in the body of that particular diseased person. So these are the different methods, but the gene therapy one is a permanent solution for this disease. Lastly, let us look at it how in molecular diagnosis the recombinant DNA technology is used, mainly to detect any disease at early stage. Molecular diagnosis is used, whether it is PCR, ELISA, or recombinant DNA technology. Now, in recombinant DNA technology, what is done? A radioactive probe is used. In PCR, a small amount can be detected by amplifying the product. And as ELISA, we know it is antigen-antibody interaction. So similarly, this can be identified by detecting antibodies or detecting antigens.